other special culture techniques. This one will be quite interesting for you all. These are quite common bacteria, but for example, Microbial leprae is cultured in the past. It's grown in armadillos. Okay, that's the only way they can culture Microbial leprae. But lately, I think they have done tissue culture. That they are able to do it on tissue culture. But I think most of the labs still do it on armadillos. Then Treponema pallidum, which is the causative agent for syphilis, grown in rabbit testicles. Then all those obligate intracellular bacteria, you only grow them in tissue culture. Okay, so these are important pathogens, but they require specialized culture approach. Okay. Other than this, you have also covered the capnophiles, bacteria that likes CO2. So for capnophiles, for the ones that require about 3 to 5% CO2, you can use a candlelight jar. It's just a simple tin can can do. Usually we choose a large one because you need to make sure the mouth is wide enough for your hands and the plates. Okay? You place the plates inverted, put a candlelight here, and then just cap it tight. Okay? When you do that, this will extinguish, but in the process, as it's burning, you will utilize oxygen and release CO2. So you will have a suitable cabinet environment. Okay? But take note that the amount of CO2 that it can produce is about 3 to 5%. So atmospheric CO2 is actually about 0.04% now, not 0.3%, 0.04%. So 3% is actually higher than usual, but it is still not adequate for certain capnophiles. So if the capnophile that you requires the highest amount of CO2, for example up to 10%, then you require a gas generator. Okay? So you can either use an anaerobic jar. Okay? Use an anaerobic jar with a gas generator for CO2, or you can use a sealed bag. Put a gas generator there and your petri dish. So usually candlelight jar or capnophilic environments suitable for Neisseria and Campylobacter. For Neisseria, you have Neisseria meningitidis and Neisseria gonorrhea. Neisseria gonorrhea causes the STD, gonorrhea. Okay? Neisseria meningitidis causes meningitis, bacterial meningitis. We've actually covered this when you're doing microbiology research. The main one will be your plate. Okay? Right? Petri plate. Petri plate, to make sure it lasts longer, you can keep in the fridge. Other than that, you can seal it. Seal it with a Petri film. Okay? Then this is mainly to reduce water evaporation so that it lasts longer. But even then, plates usually last about 1 week, no more than 1 week. Anything longer, you need slants in bottles or deep. So why bottles are better? Because you can tighten. Then that will reduce a lot of evaporation. You will keep longer. That is about 1 month. For certain bacteria, especially spore formers, you can actually just keep it in sterile distilled water. So if you're using deep, you can overlay the culture with mineral oil. Okay. essentially when you reduce the amount of oxygen, so the metabolic activity of the bacteria slows down, the nutrients in your deep can last longer. So the whole aim is to ensure that metabolic activity of the bacteria is reduced so that the nutrients that you have last longer. So either you keep it at 4 degrees Celsius, or you add a paraffin oil on top. And finally, cryopreservation is freezing at liquid nitrogen. If you want to use freezing as an approach, quick freeze, then when you want to use it, slow thaw. Why quick freeze? When you freeze it quickly, the ice crystals are smaller, so it won't hurt their membrane cells. But when you freeze it slowly, the ice crystal form are larger, then it might actually break their cell wall and cell membrane. That's why quick freeze. How to do quick freeze? Best way is liquid nitrogen. Quick freeze by using liquid nitrogen. That means you prepare your bacteria. Usually we prepare them in glycerol broth. Okay. Usually the procedure is like this. You grow your bacteria in TSB, tryptone soy broth. Okay, you grow them, but make sure you grow them until log phase. You don't grow them too long. Okay? You want your bacteria to be preserved in a healthy condition. Okay? So in log phase, you add glycerol, then you dip into your liquid nitrogen. Usually we do this in microfusions. So the liquid nitrogen is just for quick freezing. When you store it, you usually store it at an ultra low freezer, minus 80 degrees Celsius. Then this one can last for a few years. So this is for cryopreservation. We add glycerol so that glycerol is kind of a cryoprotectant. It helps to protect the cells during the freezing process. So the other one is freeze drying or lyophilization. So this requires a special equipment, a freeze dryer. Here, you remove the liquid also, so that the bacteria is in suspended animation, which means it's hibernation. It doesn't have any water molecules for it to carry out any process. That's why lyophilization can last longer than freezing. Okay? Lyophilization can last longer than freezing because it also removes the water. Many species of bacteria remain viable and unchanged in their characteristics for more than 30 years. Okay? So this is for really long-term preservation. Usually when we are doing research in microbiology, we will have a few cultures of a few types of the same cultures, which means we will have plate culture, we will have slants, and we will have glycerol stocks and all that. Okay. So the glycerol stocks are for long-term preservation, then the plate is for regular work, and the slants are usually as a insurance. Sometimes when you're working with your plates, it gets contaminated, then you still have your slants where you can re-isolate from. Okay? So the slant is more of an insurance, the plate is your working culture. So usually in microbiology research, we will always have a working culture where we keep using the bacteria, and also stock culture. So stock culture can be in the form of slants, can be in the form of glycerol stocks. But if you are really certain you want to keep the bacteria for a long time, then you can do freeze drying. We move on to the last section, I think, control of microbial growth. There are some terminology here that you should know. First is sterilization. killing all forms of microbial life in a material object or object. Okay, usually done by heat, under pressure, or sterilizing gas. You also have commercial sterilization. For example, in canned food or when you use sufficient heat treatment to kill microorganisms and endospores. So what's the difference? So sterilization, all must die. But commercial sterilization, they only focus on usually on the important ones, important bacteria. Why? Why don't they use complete sterilization even in food production? Cost is more expensive to carry out complete sterilization, which means you have to add pressure, you have to add more heat. So there's a cost involved. And second, taste. The taste of the food, the texture of the food will change. So in food industry, they can actually accommodate a certain number of bacteria as long as the bacteria is not pathogenic and as long as the numbers do not change the flavor and the texture of the food. But when it comes to Salmonella, Shigella, Vibrio, it must be zero. Because in foods, Salmonella, Shigella, Vibrio, it's either presence or absence. They don't care about the number. Okay. Next, we look at disinfection. Disinfection, the definition is destruction of vegetative pathogens. Antisepsis is destruction of vegetative pathogens on living tissue. So, one is like antiseptic, you apply on the skin, living tissue. Whereas disinfection, maybe you can wipe the table, wipe the bench, okay, wipe the apparatus to just kill. So, non-living and living tissue. This is the main difference. Then you can see the examples for antiseptic, you know. For degerming, it is removal of microbes from a limited area. Usually it is like if you have a wound and then you just remove the microbes from the wound area by swab, alcohol soaked swab. So for sanitization, the term is used usually for the utensils that you use. Okay? So the utensils that you use in the hospital, the utensils that you use in the lab have to be sanitized, have to be cleaned. Biocidal means to kill. Biostasis means to prevent growth. That's the main difference, okay? One is to kill, the other is just to prevent or inhibit growth. Then sepsis and asepsis. Sepsis is a term where there is bacterial contamination. So sometimes you hear of doctors saying blood sepsis or septicemia. Septicemia is blood infection, which means there's bacteria in your bloodstream. Okay? Then asepsis is absence of contamination. Then now rate of microbial death. So when we are carrying out sterilization in foods, they always look at how much bacteria can be removed with that process, and it's usually termed as a decimal reduction time. Using a specific process, the time required to reduce the count by 1 log. Here is written 1 log decrease is about 90% of the population killed. Then all these antimicrobial treatments, what are the factors that affect them? Number of microbes, type of microbes, and environmental influences. So of course this is very logical. The more bacteria in your initial sample, the the higher the chance that the number of bacteria left after the sterilization step, you will have bacteria left over. The types of microbes, of course, endospores are difficult to destroy. That is also logical, because even if you kill their even if you kill the cells, the spores still remain. If the spores remain, that means when the conditions are right, they can germinate. Then environmental influences, presence of organic matter tends to inhibit antimicrobials. So it seems organic matter protects bacteria. That's why when the water is very dirty with a lot of organic pollutants, the bacteria can survive chlorination. Okay? The time of exposure is also important. If this is not enough, some people will do it longer. For mode of action, so how does the chemical that you use, or how the process you use, kills the bacteria? So these are mainly antimicrobial agents mode of action. So usually mode of action can be the focus is on cell wall, cell membrane, protein and nucleic acid synthesis, protein structure and function. So for cell wall, very famous, penicillin. Most famous example of an antimicrobial agent that interferes with bacterial cell wall synthesis, okay, is your penicillin. But now we have penicillinase producing bacteria. So penicillinase producing bacteria means the bacteria can degrade penicillin. Then it will protect itself against penicillin. But you also now have beta-lactamase producing bacteria. Beta-lactam is the main structure in the penicillin antibiotic. And if a bacteria has the enzyme to to break down the ring structure, that means it can prevent the antibiotics from working. For cell membrane, usually it's the surfactants, detergents, surfactants. So you know that the cell membrane has a hydrophobic layer in between. This is to control, to regulate what goes in and what goes out of the cell. So surfactants are polar molecules that disrupts this process. So once you have surfactants that affect the cell membrane, then the cells cannot regulate what comes in and out of the cell, then it will die. So alcohol can do this. That's why when you use alcohol, you use 70% alcohol as a disinfectant, right? In your lab work. Other than that, antimicrobial agents can also affect protein and nucleic acid synthesis. Tetracycline and chloramphenicol works on this. Chloramphenicol binds to the ribosome of bacteria and then prevents protein synthesis, interrupts protein synthesis. Other than antibiotics, chemicals such as formaldehyde and ethylene oxide also interfere with DNA and RNA function. Then you can also have antimicrobial agents that alter the protein function. So protein is usually can be a 2D or 3D structure. With heat, you can denature the protein. Okay? pH change also can denature the protein, and some heavy metals can also inhibit inhibit the protein activity by blocking the active sites. Okay, so you have examples here. Metallic ions, usually copper, copper, silver also. So the chemicals, antimicrobial agents used, they affect mainly the cell wall, then cell membrane, then protein synthesis, DNA synthesis, then protein function. Okay, so these are all the places where the antimicrobial agent can act upon. Next, physical methods of microbial control. So other than chemical methods of microbial control, we also use physical methods of microbial control. Heat is the main one. So when it comes to heat, there are several terms that you have to know. Thermal death point, the lowest temperature at which all the microbes in the liquid suspension will be killed in 10 minutes. Thermal death time, minimal length of time in which all bacteria will be killed at a given temperature. Then decimal reduction time, the D value. So the time in minutes in which 90% of the population of bacteria at a given temperature will be killed. So usually used in the canning industry. So canning industry, the main focus is on Clostridium, either Clostridium botulinum or Clostridium tetani. Both anaerobic spore formers. Okay? Both environmental soil bacteria. So these two were often problems in the canning industry. So in the past, canning industry is like a household industry, which means you prepare your own cans and you sell. Okay? But sometimes the process isn't done properly and there is contamination. And people die from eating all these contaminated cans. If you look at the canned food, it is just a suitable environment for the bacteria. It's anaerobic, it's enclosed. Right? So any contamination with Clostridium botulinum or Clostridium tetani, the bacteria will grow. So when you talk about heat to kill the bacteria, there's moist heat and there's dry heat. Moist heat is more effective than dry heat, okay, because it can enter the cell better. So these are forms of heat. For dry heat, we have dry oven. An oven at 150 to 180 degrees for 2 to 4 hours. So we usually use oven, hot air oven for glassware. For incineration, we don't usually use other than for your loop. For moist heat, we use a lot, especially steam under pressure, autoclave pressure cooker, 121 degrees Celsius for 10 to 40 minutes, depending on the load size, right? So if you are utilizing a new autoclave, you must carry out tests to ensure that the autoclaving is complete. Okay? If you are autoclaving new things, the volume is different, it is best that you check it out, test it out. Okay? You must do proper controls so that you do not have problems later. Pasteurization is mainly for the food industry. Okay? You see that it's for milk and dairy products, pasteurized. I'm sure you've seen pasteurized milk. You have ultra high treatment, high temperature, ultra high temperature. You also have sterilized. But mainly the main differences is the flavor. and also the nutrients left in the milk after the different processes. So usually the manufacturer will say, if you want the highest nutrition value, you go for pasteurized, right? Because the vitamins and other factors inside are still maintained. But pasteurized milk will spoil after a few days, right? Pasteurization was invented by what did he do with the invention that made him an honorable man? Yeah, he gave it freely. He invented it mainly initially for the wine industry because the French were having problems with their wines were souring. He invented it and he gave it freely. He didn't patent it for money. He would have been very, very rich, but he didn't. So that's a very honorable thing. Okay? So now everyone remembers him from the name. Okay? So, but when when he invented pasteurization, the main aim was not to kill all the bacteria. The main aim at that time was mainly, so the organism of target in pasteurization is actually Microbacterium tuberculosis. Okay? So at that time, the organism of target when carrying out pasteurization was Microbacterium tuberculosis. Because that was a problem at that time and they do not want the bacteria the infection to spread. So all these inventions have an objective behind them. Okay? They have an objective as to why they want to carry out these steps. The sterilization is to kill all the bacteria so that the milk can last a few years. Ultra high temperature also for longer shelf life. Okay, so you have examples of moist heat and dry heat here. So you can see, most of the things are already here. The moist heat will be autoclaving, which means steam under pressure. So you've already done, for dry heat, you've already done the direct flaming, incineration. Okay, low temperature, we've already done. We say freezing will. So freezing does not necessarily kill, but may kill or may reduce the microbial population. But freezing is mainly to slow down the microbial activity, to preserve the suitability of the food, the taste of the food, the nature, the texture of the food, just to prolong, okay, to preserve the food quality. But remember, there are psychophiles. So desiccation, drying. So certain things that can be dried, food stuff that can be dried, then you keep them by drying. Okay. Then you have high pressure pascalization. So this is usually used in the food industry because in food industry, certain food processing, there are spores, spore formers. These are spore formers that are very difficult to kill, even by autoclaving. Okay. So usually they are not pathogens per se, so they are not pathogenic, but they just create problems in the food industry because they can grow in suitable environments. Currently, the only way to remove them from food processes in the food industry is by pascalization. You increase the temperature, you increase the pressure, and then you add temperature and increase pressure to kill. So steam and pressure, only that can kill. And the pascalization we're talking about is high, 100 to 800 megapascals. Osmotic pressure is more for water activity. So the concept of water activity, when you pickle, when you preserve with sugar, when you preserve with salt, essentially what you're doing is you're reducing the water activity. Because when you add lots of sugar and salts to a liquid solution, the salts and sugars will bind the water molecule. When they bind the water molecule, the amount of water molecule left for the microbes is reduced. So that is the term water activity. So pickling, preserving by sugar and salt are all based on this concept. To reduce water activity to such a level that the bacteria cannot grow. Even yeast, fungi cannot grow. For radiation, it's usually gamma rays, usually for glassware or during the industrial manufacturing. Sometimes if they don't use ethylene oxide, they use gamma irradiated petri dish. So when you buy petri dishes, you can check, is it irradiated? Is it ethylene oxide treated? You have different types of petri dishes, sterilized differently. Okay. So essentially, what non-radiation is to destroy the DNA by forming thymidine dimers, then prevents the microbes from reproducing, from replicating the DNA. So you can see that most of this irradiation is used in sterile gloves, intravenous tubing, latex and other stuff so when it's not suitable to use chemicals, when it's not suitable to use heat, then you use irradiation. Okay? So different kinds of approaches. So filtration from the pore size can remove the bacteria. So filtration is also used in the laminar flow that you use, the clean hood. Sometimes they have HEPA filters, high efficiency particulate air filters. For chemical methods of microbial control, these are used to control the growth of microbes on both living tissue and inanimate objects, and the antimicrobial chemicals can also occur in liquid, gaseous or solid state, and they can serve as disinfectants, antiseptics, sterilants, degermers or preservatives. So agents that actually kill has a suffix "cidal", for example bactericidal, fungicidal and viricidal, whereas agents that do not kill but only inhibit growth have the suffix "static", for example bacteriostatic, fungistatic and viristatic compounds. The desirable qualities in a germicide includes rapid action in low concentrations, solubility, broad spectrum, able to penetrate inanimate surfaces, able to withstand inactivation by organic matter. They are non-corrosive, non-staining, has sanitizing and deodorizing properties, is affordable and readily available. So these are all the desirable qualities in a germicide. In a hospital setting, germicides are evaluated according to their effectiveness in destroying microbes, and there are 3 levels of chemical decontamination: low levels of disinfection, to eliminate only vegetative microbes and used to clean materials, for example, electrodes, straps, and fixtures that touch the skin surface but not the mucous membrane. We also have intermediate level, where the germicides can kill fungal spores, resistant pathogens and viruses, and is used to disinfect items that come into intimate contact with mucous membranes but are non-invasive. For example, thermometers in the mouth. And finally, high level, they are able to kill bacterial endospores if properly used as sterilants, and are used for medical devices that are not heat sterilizable, for example, catheters, endoscopes, and implants. Then the effectiveness of these agents depend on the numbers and kinds of microbes that are present, the kind of materials that are being treated, time of exposure required, the strength and mode of action of the agent. To follow the effects of antimicrobial agents on growth, usually we carry out a time course experiment where we follow the bacterial counts, cell numbers with time. So enumeration of bacteria, we can use epifluorescence microscope or hemocytometer for total direct count, total cell count, or we can use the culture approach for viable cell count. So if we were to follow the cell number over a period of time, if the agent is added and then we do not see any increase, and when we remove the agent it increases, then the chemical is a bacteriostatic in nature. However, if after adding the agent the viable cell count drops and the total cell count remains as it is, then it is bactericidal because the cells died. Okay, what we observe here are just the non-living cells. For bacteriolytic is when the agent causes not only the death of the bacteria but also bacteria lyse. So there is no cell count. The cell count also decreases. Okay, so this is the different patterns that we should observe with different types of agents. And to assess antimicrobial activity for any chemical that you have, we can carry out a minimum inhibitory concentration assay where the chemical agent is diluted using zero dilution, and then each tube is inoculated. The tube with the lowest concentration of antimicrobial agent that doesn't have any growth is the MIC, minimum inhibitory concentration. So this table shows the types of common antiseptics, sterilants, disinfectants, and sanitizers used in the hospital environments. So we have covered the different types of microbial control methods, where for physical agents we looked at heat, and for heat we looked at the dry heat and the moist heat. Other than heat, radiation, it was also a classified under physical agents, and we have ionizing and non-ionizing rays. For the mechanical removal methods, the focus is on filtration, and then for chemical agents we have examples of gases and liquids that can be used to sterilize and disinfect materials. Finally, for microbial metabolism, know that the metabolism is all chemical reactions that occur within a living organism and can be generally divided into catabolic and anabolic processes. Catabolic is degradative, breaking down, and usually coupled to ATP synthesis, energy producing, whereas for anabolic, it is usually to build more complex molecules from simpler ones and usually require energy. So these two processes, catabolic and anabolic processes, are coupled because of the requirement of energy here and the generation of energy here.